Good morning, students. Hope you will find at your respective places. Last time we have discussed this that this nitrogen fixation it can take place by two methods. First one is the physical nitrogen fixation, and second one is this biological nitrogen fixation. Last time we have discussed this physical nitrogen fixation where we have discussed it in two categories, that is this natural nitrogen fixation and second one is the industrial nitrogen fixation. Today we are going to discuss this biological nitrogen fixation, and this can be further classified into two categories, into two headings. This biological nitrogen fixation, it can be discussed into two headings. First one is called as non-symbiotic non-symbiotic nitrogen fixation, and second one is symbiotic nitrogen fixation. Symbiotic nitrogen fixation. So this biological nitrogen fixation, it can be discussed under two headings: non-symbiotic nitrogen fixation, and we have got the second one that is called as this symbiotic nitrogen fixation. Now what happens in this non-symbiotic nitrogen fixation? This non-symbiotic nitrogen fixation, it is shown by many aerobic and anaerobic. This non-symbiotic nitrogen it is shown by many aerobic and non-aerobic bacteria. Then we have got this blue-green alga, which is called as cyanobacteria. Then it is shown by this fungi, etc. So this aerobic and non-aerobic bacteria, and also by this blue-green algae and fungi. Now I will give the examples of this non-symbiotic nitrogen fixation of the animals which show this. पहले aerobic bacteria, AEROBIC. अगर हम aerobic bacteria की बात कर लेंगे, it is shown by this azotobacter. Then we have got anaerobic bacteria. It is shown by Clostridium. Then we have got a blue-green alga. Yeah, we can say it by another name, cyanobacteria. For example, we have got nostoc. For example, we have got this anabina. Then we have got this fungi, and this fungi में अगर हम देखेंगे we shows this non-symbiotic nitrogen fixation, that is called as thiobacillus. No, sorry, that is shown by this yeast cells. Yeast cells, where we will see this nitrogen fixation can take place. Now, what happens in this whole process in this non-symbiotic nitrogen fixation? See what happens in this process. So, we were discussing this non-symbiotic nitrogen fixation. So what happens in this process? Huh? Again, this process, the process, it is what we'll see, जो हम इसमें देखेंगे, nitrate assimilation, that is nitrate assimilation, nitrate assimilation. So what happens in this process, whole process, जो nitrate जो होता है, कौन, जो नाइट्रेट होता है, नाइट्रेट रिडक्शन, जो नाइट्रेट, इट इज रिड्यूस्ड टू अमोनिया, this nitrate, it is reduced to ammonia, and this whole process, it occurs in the following steps. Guys, see, I will write this. Nitrate, it gets converted into nitrite. Nitrite जो है, it gets converted into hyponitrite. Hyponitrite, it gets converted into hydroxylamine. This hydroxylamine, it gets converted into ammonia. So what happens in this case, जो nitrate होता है, it gets reduced to ammonia in the following steps. Nitrate, it gets converted into nitrite. Nitrite gets converted into hyponitrite. Hyponitrite gets converted into hydroxylamine, and hydroxylamine gets converted into ammonia. And it takes place in two steps. What are those two steps? पहला है step first. What is that step first? That is a nitrate reduction. That is this first step. So what we will see in this first step, nitrate is reduced to nitrite. Nitrate is reduced to nitrite by an enzyme. What is that enzyme known as? That is called as nitrate reductase. Nitrate is reduced to nitrite by an enzyme, nitrate reductase. This is an enzyme which reduces this nitrate into nitrite. Now, अगर हम इसका स्ट्रक्चर देखेंगे, इस नाइट्रेट रिडक्टेज का, दिस एंजाइम इज सिंपली, दिस एंजाइम, दिस इज फ्लेवोप्रोटीन. This nitrate reductase, it is simply a flavoprotein, and it contains FAD. It contains FAD. What does that full form mean? Flavin, 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 adenine, diphosphate, dinucleotide. So this nitrate reductase की अगर हम बात कर लेंगे, it is a flavoprotein which contains FAD, that is flavin adenine dinucleotide, as prosthetic group, and receives hydrogen from, hydrogen कहां से हासिल कर लेता है? From reduced and received received hydrogen from reduced NADP or NAD. Now, in this nitrate reductase, there is the molybdenum. In this nitrate reductase, there is molybdenum, and this molybdenum it acts as enzyme. It acts as electron carrier. So in first step, what we have to discuss this nitrate reductase. So what happens? First, this nitrate it is reduced to nitrite by an enzyme nitrate reductase. Now this nitrate reductase it is a flavoprotein which contains FAD, flavin adenine dinucleotide as prosthetic group and which receives hydrogen from which receives yeah hydrogen from reduced NADP or NAD. Now, in this enzyme, there is molybdenum, and this molybdenum in the enzyme it acts as electron carrier. This molybdenum it acts as an electron carrier. So first step जो हमने किया, we have reduced this nitrate into nitrite by means of this what we call as this enzyme nitrate reductase. Step second, अब हमारे पास नाइट्राइट आ गया. Step second, so what is this step second? It is that is nitrite reduction. Now what happens in this reaction? जो nitrite ion जो होते हैं, they are reduced to ammonia. So nitrite ion जो हैं, they are nitrite ions. They are reduced to ammonia by means of an enzyme, and इस enzyme का नाम क्या है? That is called as nitrite reductase. By means of enzyme nitrite reductase. These nitrite ion they are reduced to ammonia by means of this enzyme nitrite reductase. Now this whole process, this nitrite ions when they get converted into ammonia, it takes place, this change occurs in leaves. This whole process it takes place in leaves in presence of light. In presence of light, जब हम लाइट में देखते हैं कि दैट होल प्रोसेस इन लाइट इट हैपन्स वेरी मच क्विकली, बट इन डार्क लाइट जो स्पीड है इसकी रिएक्शन की वो थोड़ी बहुत धीमी हो जाती है. So this is due to this reducing power of reaction from photochemical splitting of water. Why this? हम ये कहेंगे this nitrite ion it gets reduced to ammonia by means of this nitrite reductase, and it mainly happens in the leaves, and जो इसकी speed होती है that is very much high in the light. It is because it is due to the reducing power. It is due to the reducing power of reaction. It is due to reducing power of reaction from, हम देखते हैं, photochemical splitting of water. When in the light this water in the leaves it gets broken down, and during this process whole this nitrate ion it gets converted into the ammonia. So nitrate HNO3, अगर हम रिएक्शन लिखेंगे, this HNO2 plus H2O, it gives us ammonia, NH3 plus oxygen. So this nitrite ion HNO2 plus water, it gets converted into ammonia and oxygen. Now in first case where we will see step first में नाइट्रेट रिडक्शन जब हम क्या देखते हैं कि देयर वो नीड द मॉलिब्डेनम, लेकिन यहाँ पर हम क्या देखते हैं कि दिस नाइट्रेट रिडक्टेज इट डज नॉट नीड दिस मॉलिब्डेनम, यहाँ पर हमको मॉलिब्डेनम की जरूरत नहीं पड़ती है, बट इट मे रिक्वायर द प्रेजेंस ऑफ आयरन, इट मे रिक्वायर द प्रेजेंस ऑफ कॉपर, कि ये नाइट्रेट रिडक्टेज इसको नाइट्राइट आयन को रिड्यूस करने के लिए अमोनिया के लिए हमको मॉलिब्डेनम की जरूरत नहीं पड़ती है, बट वी नीड दिस आयरन एंड वी नीड दिस कॉपर, एंड सिंपली NaDH and NADPH and NADPH, they act as hydrogen donors. They act as हाइड्रोजन डोनर्स, दे एक्ट एज हाइड्रोजन डोनर, उनका काम सिर्फ क्या है, हाइड्रोजन डोनर्स का. सो दिस वाज ऑल अबाउट दिस बायोलॉजिकल नाइट्रोजन फिक्सेशन, लेकिन ये क्या है, नॉन सिम्बायोटिक नाइट्रोजन फिक्सेशन में, एंड इट इज शोन बाय वेरियस एरोबिक एंड नॉन एरोबिक दीज बैक्टीरिया, इट इज शोन बाय द साइनोबैक्टीरिया, इट इज शोन बाय द फंगस, एंड होल ऑफ दिस प्रोसेस इट टेक्स प्लेस इनटू टू स्टेप्स. फर्स्ट वन इज द नाइट्रेट एसिमिलेशन, नाइट्रेट रिडक्शन. नाइट्रेट रिडक्शन में क्या हम देखते हैं कि जो नाइट्रेट जो होता है, it is reduced to the nitrite by means of this enzyme nitrate reductase, and this what we will see molybdenum in the enzyme, it acts as an what we call as this electron carrier. Second step में हम क्या देखते हैं कि जो नाइट्राइट आयन्स होते हैं they get convert, they are reduced to the ammonia by means of the nitrite reductase, लेकिन यहाँ पर हमको molybdenum की जरूरत नहीं पड़ती इस एंजाइम में, लेकिन it needs the iron and the copper, and NADH and NADPH, they act as the hydrogen ion. Now whatever this nitrite ion they get converted to the ammonia, they takes place in the leaves in presence of the light. वहां पर हम क्या देखते हैं कि लाइट में ये इसकी स्पीड रिडक्शन स्पीड जो है दैट इज समवट फास्टर एज कंपेयर टू दैट ऑफ द डार्क. उसकी वजह यही है, दिस इज ड्यू टू द रिड्यूसिंग पावर ऑफ रिएक्शन फ्रॉम फोटोकेमिकल स्प्लिटिंग ऑफ वाटर, एंड द होल रिएक्शन जो हमारे पास है दैट इज दिस रिएक्शन नाइट्राइट आयंस it gets converted into the ammonia, and there we will see this water जो यहाँ पर है it splits, that takes place in presence of the sunlight, that is why it is called as photochemical splitting यहाँ पर हो जाती है, and oxygen is evolved. So this was all about this non-symbiotic nitrogen fixation. In the next video we will try to discuss this symbiotic nitrogen fixation. Until then, goodbye, God bless you, thanks.